All right, so like I said, we're entering into a brand new phase of the course. We've gotten through population genetics, we've talked about evolution, we talked about allelic frequency changes. And that set the sort of the stage for everything we're going to talk about now. Now we're moving into the diversity of life. We talked about how life began. Now we're going to talk about how it's diversified over time. So today we're going to begin with a discussion of prokaryotic cells. We're going to need to be able to identify major phylogenetic groups, use examples to characterize the supergroups of eukaryotes, describe characteristics of organisms under domain bacteria and archaea, and recognize representatives of prokaryotes and protists. So we're going to play a game later called Is it a protist or a prokaryote? This is wrapped into that diversity and evolution part of the course. We need to look at the wider range of life living right now. And that's why this part is so cool, because all of the things we're going to be talking about from now on are things that exist around you. Prokaryotes have been around since the beginning. We ended the last lecture talking about simple cells, those protobionts, the RNA wrapped in a liposome. Now we're moving into much more complex organisms. Now, complex is sort of a relative term. Much more evolved organisms. For billions of years, life has been changing, one generation after the next after the next. And it's branched. This is, I guess you'd call it, the phylogenetic tree of all life. And you'll notice we're right back to those three domains that you started Bio 101 with: bacteria, archaea, and eukaryota. What I want you to notice from this image is that prokaryotes are going to be paraphyletic. That means that they have a common ancestor, but it does not include all of the descendants. These eukaryotes are off on their own. So archaeans actually end up really being a lot closer related to eukaryotes, even though they're prokaryotic. So here are all of the groups of bacteria, and you'll notice that they had a split, and then way over here are the eukaryotes and the archaeans. You have the eukaryotic group over here, and the prokaryotic group here, and then all of these other ones. There's a lot of evidence to show, not that there's a direct relationship between archaeans and eukaryotes, but that eukaryotes are actually a combination of bacteria and archaea. I'm going to get into each of those. You guys know what a bacteria is, right? You've at least thought about them. At some point in your life, you've coughed and you thought, maybe I've got a bacterial infection. And there's some of you who, when you get sick, you're like, I hope I have a bacterial infection, because it's a virus, I just have to deal with it. And if it's a bacteria, I can go to the doctor and they'll give me something and it'll go away. On Earth, they're an estimated lots. Okay, hold on. I can work out this number. So we got 1,000, million, billion, trillion, quadrillion, pentillion, hexillion, septillion, octillion, nonillion. One nonillion bacteria at any given point on the Earth. That is half of all the biomass. We don't see them, but they weigh as much as everything else. Like literally, there's prokaryotes, and then everything else. You don't see the prokaryotes with your naked eye, but they provide that much weight. Heck, we're going to talk later that roughly a quarter to half of the weight of your feces is dead bacterial cells. And you guys know how much you excrete daily. I don't want to know, but you guys know. If you were to go outside and take a pinch of soil, and that's it's like your grandmother's recipes, take a pinch, literal pinch. You go like this, pinch the soil. What is in your fingers has as many bacteria as there are humans in the world, about 4 to 8 billion. That's a lot of bacteria in your fingers. And then you need to wash your hands. In case you didn't know, super important to continually wash your hands. So that got me thinking, because it was late when I wrote this lecture, like really late. And I was like, what if, you know the story Horton Hears a Who? All right, The Who's that live in Whoville, they don't live in Whoville. That's where the, are they the same? Hold on. Horton here's a Who.
Well, I think it's the same concept.
It's the same How do we know that? No, seriously. Okay, sidebar. We got to talk about this.
The design between the two towns are radically different if I remember.
Yeah, but that could be a population. So, okay, this is just blowing my mind now. Are the Who's from The Grinch Who Stole Christmas just really, really small? They're living in their own little tiny microscopic world.
Well, again,
They are.
the beginning of
He's dead.
The beginning of the Grinch actually starts with zooming in on a snowflake.
But does the book start that way? Or
I don't think so.
that could be just that's.
But it's the original animated movie, which was influenced by Susan stuff.
All right, guys, this is going to be important to look up later. Yeah, sidebar. I have no idea what I'm talking about now. Okay, whatever. I don't care anymore. Who's are really small. Prokaryotes are really small. Moving on. Prokaryotes. So I have to delete that last slide. Prokaryotes are in every possible environment that we know of. Everywhere we have looked on Earth, we have found prokaryotes. Every place that you would look at and it's like, nothing can live there. There are bacteria, there are Archaeans or bacteria living there. Whether in glaciers, deep sea vents, heck, thermal features out in Yellowstone that nothing else survives, and you've got bacteria there. They're all over the place. Some people turn around and they say, well, Archaeans and bacteria, they're obviously the most ancient lineages. They are the oldest organisms. You've probably heard that. I don't like that idea. And here's why. And I think your book actually says something along that line. I don't like that idea, and here's why. If you look back at that phylogenetic tree, right, it all stems from one last universal common ancestor. That means that as those bacteria diverged and became what they are now, we diverged and became what we are now. All of these lineages came about from a single source. So how can any of them have been one older than the other? Now, it all comes down to taxonomy, and as we recognize, as I've said many times in this course, taxonomy is dumb. You could trace phylogenies back, figure out what's more evolved than anything else, but in the end, what that's just going to tell you is exactly what you wanted to find out in the first place, whatever you think. It's all based on what you, the scientists, think. I did put this image up. This is a bed of seagrass. People talk a lot about what's the oldest animal, what's the oldest organism. Turns out this bed of seagrass has been around for thousands and thousands and thousands of years, and it's the same genetic structure. So this grass is thousands of years old. There are trees out there that are like 1,500, 1,600 years old. Sadly enough, just this summer, somebody went and cut one of them down. They just went and cut this ancient tree down that almost predated human civilization. And they're like, good, it's got to go. People are stupid. Takeaway message, people are stupid. Over billions and billions of years, bacteria and archaeans have established a lot of metabolic diversity. If there's a substrate out there, it can be broken down by a bacteria or an archaean. They will use anything. There's something out there that will break it down. The reason this occurred is because both bacteria and archaeans have a high reproductive rate and a high mutation rate. As we learned in previous lectures, that increases the speed of evolution. The other one, harkening back to your exam, is of course allopatry, having a barrier come up in the middle. But that high mutation rate and the high reproductive rate can really, really cause the genes of the prokaryotes to change quickly, which allows selection to occur based on their environment. Now here's the deal. We keep talking about these prokaryotes as if they're one big group, but they're not. There's two distinct lineages, the Archaeans and the bacteria. Here are the differences between them. And at this point, you've taken one of my exams. And you know I'm super interested in things like, what are differences between these? What are similarities? Well, here's the deal. Archaeans have histones. If you remember back to Bio 101, a histone is a protein that DNA wraps around in order to create chromosomes. Bacteria lack histones. So here's a way that Archaeans are very similar to eukaryotes and very different from Archaeans. I'm sorry, yeah, different from bacteria. Archaeans have an extra ribosomal protein. So ribosome, what are ribosomes good for? Do you guys remember? Building proteins, building polypeptides. So here we have I know you guys aren't terribly concerned with protein structure, but hear me out. A is what a bacteria's ribosome looks like. B is what an Archaean ribosome looks like. It has this whole extra piece that sort of fits inside. This Archaean ribosome looks almost exactly like a eukaryotic ribosome. So here's where, again, bacteria and Archaeans are different. But Archaeans and eukaryotes are similar. If you look at the RNA polymerase, gosh, I'm back to protein structures with you guys, I'm sorry. You've got a bacteria's RNA polymerase. What does RNA polymerase do? Do you guys remember from 101? I'll help. It builds RNA. Yes, it builds RNA. It polymerizes RNA. So it takes individual nucleotides and builds an RNA molecule out of it. So here's what it looks like in bacteria. You've got the sort of U shape here. But when you look at an Archaean, it looks like you've got that U shape still, but you got these extra pieces that, I guess, make it look sort of like a swan. Down here, you've got bacteria, you've got Archaeans, and you've got eukaryotes. And you'll notice, again, oh, it happens all the time. Archaeans look a lot like eukaryotes. So I was just talking to another professor that said, why do you talk this much depth with them about Archaeans and bacteria? Well, the reason I go into this much depth is because it's not only what sets them apart, but it's what allows Archaeans to survive environments everything else dies in. This is a huge issue. Archaeans have ether-bonded phospholipids. So their phospholipids have cross-linkages. You guys remember what a phospholipid is, right? Oh, please tell me you do. Okay, good. A phospholipid in most cells, eukaryotes and bacteria, have what are called ester bonds. There's an S instead of a T. They're not linked to each other. But in bacteria, not only do they go next to each other, in Archaeans, they not only go next to each other, they hold on to each other. It's the difference between having a bunch of random boards stuck in the ground. Got it. When you were younger, did you ever make a fort out of popsicle sticks? Do you have any idea what I'm talking about? No, you guys never made a fort out of popsicle sticks? Okay.
You mean like a house?
Or like a house out of you take popsicle sticks and you build something. How about that? Okay. No, I'm not telling like something you hide in. Just like a tiny house for a project. One way you could do it is you could take individual popsicle sticks and glue them down one by one, right? And they could stick to the cardboard. That's not very strong, and it may blow down. It may fall down. Yeah, big bad wolf comes along. I don't know. Another way of doing it would be to take those pieces and stick them down, and then put a popsicle stick behind them. That creates a structure. So you could have them sort of just sticking in like this into some material. So you've got a bunch of sticks sticking straight up. This is what bacteria and eukaryotes have. But what Archaeans have is they've got that same basic structure with an extra support mechanism. So it holds them together, prevents them from falling apart, even at super high temperatures, even in super acidic conditions. So these ether bonds are resistant to heat. They're resistant to damage. When a cell might otherwise collapse, they can survive. And that's what lets Archaeans do some super amazing stuff. Archaeans are what are known as extremophiles. I'm not kidding, real word. They're extremophiles. Somebody put it that they're the Mountain Dew drinkers of the bacterial world. They survive in extreme conditions. They can do that because of their unique bonds of their phospholipids. They might be acidophiles, all right? Where do you think they live if they're called an acidophile? Right, acid. Acido, acid. Phile means love, so they love acid. These organisms thrive at pH levels of 3 or below. So 3 or below, they're doing really well. Super acidic, lots of hydrogen ions. There are pits open in the earth in which there is super acidic, super highly heated acid. Archaeans live there. You will die there. Heck, if you go to Yellowstone National Park, you can walk along the boardwalk, and they say stay on the boardwalk, because if you walk off the boardwalk, there are pits of acid that's boiling hot that you can fall into, and people have, because people are dumb. Back to our thesis of the day. People are dumb. Yep. So you might be an alkalophile, an organism with optimal pH growth at 9 or above. They love alkaline environments. Think mine tillings, right outside of a mine. In case you didn't know, mines can damage ecosystems like crazy. They're going to be organisms that survive in that mine runoff. They're not the kind that you can eat. They're not the kind that you want to have really surviving there, but they'll survive. Because in the end, life finds a way. It's going to be the Jurassic Park, right? Life will find a way. Right, we may not survive, but life will. Cryptoendoliths, which is a great Scrabble word. The cryptoendolith are organisms that live in microscopic spaces in rocks, where little tiny bits of water can get in there. In between the grains of soil, life can live. They might live in aquifers, underground. You can dig deep down. Anywhere water goes, you'll have bacteria, some of which are going to be cryptoendoliths. Halophiles, they love salt. That's all I got on them. There are entire areas of the world that are what are known as salt basins. They happened because if you trap salt water, it will eventually evaporate, leaving behind the salt. I don't know if you guys have ever left a glass out and seen like around the rim sort of these crystals forming. That happens in salt basins all the time. In fact, the Mediterranean Sea, you guys know where the Mediterranean is? Go, geography. In the Mediterranean, there's an area where it's called the Straits of Gibraltar. You know what I'm talking about there? All right. That area is continually dredged because sediment keeps building up there, threatening to cut off the Mediterranean. So if they don't keep dredging it, that means that water will stop coming in from the Atlantic Ocean. So far, so good? If that happens, if that dredging stops and the water's cut off, what will happen to the water in the Mediterranean? It will slowly evaporate, leaving behind a giant salt pan. That will dramatically change the environment of every Mediterranean nation. Mainly, you're going to have a desert where there used to be an ocean. Hyperthermophiles. Fun. Hyper, lots. Thermo, heat. Phile, love. They love it where it's really hot. 80 to 122 degrees. Down at the hydrothermal vent systems. Heck, there's some bacteria that survive in excess of 400 degrees Celsius, 4 times boiling. They love the heat. A hypolith, They live underneath rocks and in cold deserts. So deserts have no water, really, no moisture. These hypoliths are going to live underneath the rocks there. So they're not baked in the sun, but they survive with very low water. Lithoautotrophs. Their sole source of carbon dioxide is from exergonic. What does exergonic mean? Energy is released. Inorganic, oxidation. So molecules that are not made by organisms, so they're not things that you could readily eat. Think rocks, right? What they do is they take rocks, they oxidize them. Man, so many stupid Bio 101 terms. Oxidize means what? Okay, what's oxidation? How about that? Remember that Leo goes Gur thing? I'm sorry, say. Right. So if you gain electrons, you're reduced. If you steal electrons or you lose electrons, you're oxidated. So these guys, what they're doing is they're oxidizing inorganic rocks. They are stealing electrons. Huge biology note. And it's going to sort of run across all of the stuff that we're doing for the course. We get our energy from the electrons we steal. The way that energy moves through any biological system is oxidation. You oxidize a substrate, you steal its electron, and it does something. When you die, your parts are oxidized in order for something else to use that energy. Oxidation is what moves energy through a system. We'll hit that a lot, especially in ecology. So these bacteria are going to oxidize substrates. They steal electrons from these rocks. They get energy from it, and those rocks break down. This is one way we start to get soil. These were one of the earlier organisms on the surface of the Earth. Metallotolerant. They're capable of tolerating high levels of dissolved heavy metals. So these guys survive in areas where we would otherwise die because we cannot tolerate those heavy metals. In fact, they were doing a sampling with this gentleman here. Why I love this picture. He did a sampling of this pool filled with heavy metals, and he dipped his bucket in. The bucket didn't survive. Literally, it completely dissolved in that solution. However, there are plenty of bacteria surviving in there. A metal bucket didn't make it, but the bacteria did. That just blows my mind. Osmophiles. This is true. This is interesting. They are organisms capable of growing in a high sugar concentration. There are organisms adapted to growing in high amounts of sugar. Literally, if you were to take a bag of sugar, a bacteria could live just in there. Now, the reason that's interesting is because sugar, salt, all of these make a, what's it called? I got phyllic stuck in my head. Hypertonic condition. All right, salt, right? What if you put salt onto a slug? What happens to the slug? It dies. It shrivels up because all the water leaves it. We used to pack food in salt and in sugar. Heck, we still do it. You guys have seen like those sugared sugared fruits, like I don't know, ginger with a lot of sugar on it. The reason they do that is to pull all the liquid out. Meats were packed in salt to pull the liquid out, because without liquid, bacteria don't survive. But these guys could. Super high sugar concentrations. Piezophiles. They live deep, deep under the sea, super high pressure. Oceanic trenches. You've probably seen videos and such. The idea is right now we have a lot of air pressure pushing down on us. We call it 1 atmosphere's worth. You can feel it. As you dive into the water, you're at the top of the water column. You don't feel too much pressure. You can feel water around you though, right? You've been swimming before. The deeper you go, the more pressure there is on you. It pushes against you. As you go deeper and deeper, what ends up happening is the air in your lungs begins to contract. It begins to condense. It gets really tight. Your lungs get smaller. You can't hold your breath that well. You keep going down. Eventually, you will be unable to breathe even if you have a scuba tank on you. There's a maximum limit humans can go down underwater, unaided, without a submarine. Does that make sense? Then we've got these big submarines. Submarines, you've seen them. They're big and they're giant and they're metal. Do they have terribly many windows on them? No, because what will happen is the deeper you go, the difference in pressure from the outside to the inside will push those windows in. It'll crush, as you go even deeper, it'll crush a sub. All this big metal infrastructure, the pressure is strong enough to condense it down. Sperm whales dive deep. They've got gigantic lungs. You guys have an idea of how big a sperm whale is, right? They're huge. They've got huge whale-like lungs. They come to the surface and they take several deep breaths. They come up. You've probably seen blowhole stuff comes out of it. They take a deep breath in. They do that several times. They blow out, they breathe in, they blow out, they breathe in, and then you'll see their tail come up as they dive straight down. As they're going down, their lungs begin to shrink more and more and more. They had to take those deep breaths so they could have that much oxygen in there. When they get into their deepest dives, their lungs are about the size of a volleyball because of the compression. Yet there are bacteria that survive at these depths where no other organism can survive. You know what else survives down there? This is interesting. The blobfish. Have you guys ever seen a blobfish? That's super ugly on the surface because it's used to super high pressure. When it's underwater, at that pressure, it looks like any other fish. It's just pressure does weird things. Psychryophiles and cryophiles. They survive at super low temperatures, below freezing. That's interesting because cytoplasm or cytosol is aqueous, which means it should freeze. But at that temperature, these guys are able to survive. We have the xerophile. The xerophile grows in very, very hot, dry conditions. Makes sense? And the final one, the polyextremophile. They don't like just one of these conditions. They like multiple conditions. They may like the ones out in Yellowstone, super acidic, and super hot. Multiple categories. So, big takeaway is every single environment that we know of on Earth has provides sustenance to some sort of prokaryote. Everywhere. We cannot look anywhere without finding them. These Archaeans at least, are very closely related to us. But they still lack that true nucleus. They still lack compartmentalization, or at least true compartmentalization. The trick now becomes, we see it everywhere on Earth. Is it possible for life to arise elsewhere? And this is where we get into a little bit of speculation, but the more we're looking at the data, the more it seems like there are likely early life on the other planets that we've explored, just Mars. There's some indication that there was at some point early life. Maybe not complex life like multicellular life, but at least those early liposomes. Mars, in fact, has regions in its deep subsurface with permafrost. Permafrost is cold and water. This could harbor cryptoendolith communities. We haven't dug down deep into them yet. Heck, the subsurface water of Europa. Europa is one of Jupiter's moons. Europa has active geothermal vents. And one thing we learned about here, and not just active geothermal vents in water. If life happened here at the vents, it could happen there at the vents. We just can't dig down deep yet. So there's this whole look for, let's push to find intelligent life out in the stars, and that's fine, but it's more likely that what we're going to find is primitive life. And it doesn't just have to be in what we call this Goldilocks zone that we live in. So if we find life external to Earth, it's probably going to look a lot like the Archaeans.